Great. Hello, testing. Hi, everybody. I'm Jason Key from Harvard Medical School. Thank you for coming for the research computing session. We're going to kick it off with a short talk from Glencoe. Aaron Deal, Glencoe is a sponsor of this session. And then I'll follow it up with a talk about research computing software. So.
Thanks everyone for joining for this session. So I'm Erin Deal. I'm head of product for Glencoe Software. We largely do image data management. And so I know there may be some people working with image data in this crowd, maybe some people not working with image data. So I'll introduce you to what we do as a company, our main data management solution, and then we have a new product coming out, kind of a new tier of an existing product that leverages some infrastructure as code. And so I thought it may be an interesting topic for this research computing session. So to start, what does Glencoe Software as a company do? I think I'll use keys here. Okay, so we aim to build the most comprehensive scientific image data management solutions, and we're doing this at a global scale. So although we are a commercial company, the software that we are building and supporting is backed by open source tools. We really like this because we find that the open source community is really rapidly evolving to meet emerging needs. But we work with some of the world's largest organizations, including things like pharmaceutical companies, but also the largest universities in the world. And so we know that we need to balance this evolution with some security and stability. And this is, of course, in parallel with our own service and support to our customers. So ultimately, when we're talking about data management, these software solutions are aiming to enable faster and easier access to your data, which we think enables you to understand your data more easily and then ultimately make greater insights from that data. So Glencoe as a company has an official basis in the UK and in the United States, but we're a fully remote team. Across that team, we're a small team, there's about 20 of us, but we have a lot of really nice varied expertise across different domains. So we end up working with a lot of scientists. So many of us are scientists by training. I was a neuroscientist in a former life. Many of us have done imaging for quite a long time in various roles. I used to work in a light microscopy facility prior to my time with Glencoe Software, so the core facility world is very near and dear to my heart. We do a lot of work with publishing. So earlier in the meeting, we did a session on publishing image data, sharing image data with our software solutions. So if that's interesting to anyone, how do we not only acquire the interesting research data, but then ultimately share it when we're ready to publish that? We have some materials at our booth and happy to have those conversations after this talk. We largely work in R&D. You'll see in some of maybe the images and slides that I have, there's certainly some use cases for more clinical workflows, but R&D is kind of the main place that we work with our customers. And of course, we have a lot of expertise in engineering because we are a software company. So our customers, I sort of mentioned earlier, they may be large pharmaceutical companies, smaller biotech companies, but also some of the world's largest universities. We really like working with academic groups to cover the varied research context that the core facilities and other labs are working on across the university. So as I mentioned, we're about 20 people, and actually this slide I failed to update, so we're actually 19. We just had a new member join just 2 weeks ago, so we're 19 now, just approaching 20 people. But of course, because we're tied with this open source project called the Open Microscopy Environment, there's many more contributors to the solutions that I'll tell you about today that are not necessarily directly associated with Glencoe Software. And Glencoe was originally founded in 2005 by 2 of the creators of the open microscopy environment. So I want to spend a little bit more time talking about this connection with the open source community because it's something that often comes up in questions about how Glencoe Software works. So some of you may be familiar with the open microscopy environment. A couple of the tools that I'll mention throughout this talk are available open source through the Open Microscopy Environment, or OME. This is a group that's still around today, grant funded, many working out of the University of Dundee. So the lead of OME, Jason Swedlow, is also the founder and still current CEO of Glencoe Software. So these 2 teams still work very closely together, and we are the exclusive commercial partner of OME. And so we really consider this a long-lasting partnership between the two groups. And so what does it mean to work with Glencoe software if many of the tools, the kind of core of our products, are available open source? The first thing to say is that we absolutely encourage people to use the open source versions of the data management solutions that I'll tell you about today. But when working with Glencoe software, some groups really find that essential to have a commercial license. So I have mentioned scale a couple of times, that these data management solutions are sometimes global solutions with large organizations, large universities. And so IT really prefers there to be some terms and conditions written down on a piece of paper in that commercial license that describes our working relationship. And so, that is just one really important difference in working with open source software compared to a commercial license with Glencoe software. But arguably on a day-to-day basis, and in particular in a core facility, probably the latter two are more essential to you. So how can we customize these deployments to reflect the actual ways that you are working with your data, the actual formats that you care about, the kinds of visualizations or analysis that you need, and the architecture that your IT or research computing departments really rely on, the kind of security requirements that you might be held within. Service and support is arguably the most important for the Glencoe software team. These data management solutions are running for many years and in some cases decades at these institutions, and so we want our solutions to grow with your research, and that means that we're really constantly communicating with our customers. We really don't deliver the software and then walk away. We really like these very direct support relationships with our customers, and so that service and support is really, really important to us. And I'll come back to that at the end of my talk in relation to this new version of Omero Plus that we have available. Okay, so what is Omero Plus? What is this data management solution that I've been kind of hinting around? So when I think about data management, as I mentioned earlier, I really think about data access. And so accessing data could mean different things. It could be viewing the data, analyzing the data, sharing it with a colleague, adding annotations or metadata to that image. And all of that becomes very hard, even just looking at an image becomes very hard when the data acquisitions are quite large. So they could be large in volume of a single image. Some domains, the acquisition that we're talking about is in the terabytes range. Certainly in the gigabytes is also quite common. But it could also be in the size of the collections. Maybe individual images are small, but you have many, many, many of them. And so looking at these becomes actually kind of complicated. We also work with a lot of proprietary file formats in bioimaging from various microscope vendors, and so this also presents a challenge in just accessing the image data and building interoperable solutions. So Omero Plus is enabling that access from anywhere you are, so remote data access, and ideally within a single software interface. And I can't really explain the way that we're able to do that without defining this tool called bioformats. So I mentioned that in bioimaging, many of you probably deal with this, there's a number of different proprietary file formats. So the microscope vendors are motivated to write data from the instrument as quickly as possible, and we're quite glad for this. But we want to, the outcome of that is that they're writing to often proprietary file formats that are not interoperable. And we want to be able to work with imaging data from across different domains and not just be stuck within a single vendor's platform. So bioformats is an image translation library. It's also available open source. It's integrated into a lot of different open source software, so you may already be working with it in a variety of contexts. And it doesn't mean that you're converting your data. It means that we've effectively reverse engineered readers for all the popular file formats in bioimaging. And what that means is that tools that use bioformats can read image data and metadata from those proprietary file formats and present them in a uniform manner. And so Omero Plus uses bioformats behind the scenes to be a vendor and domain agnostic platform. So bioformats currently supports over 160 different file formats. This is constantly growing, so some of the work that Glencoe software does is that we develop new readers for formats that our customers care about, and almost all of that work is contributed back to the open source. There are exceptions, but really only when something could be, maybe we don't fully understand it yet. We have some small set of examples of that file format, and it's not really yet supportable in the open source community, but we try and have all of that development go back into the open source because we really consider bioformats as kind of a standard of how we work with imaging data, and so it doesn't really benefit us to keep that code private to Glencoe software. And so as I said, Omero Plus uses bioformats, and that's how when I say that you can view and share and analyze your data, you can do that for all the various data types that are acquired across numerous core facilities because of a tool like bioformats and the support for various vendors' proprietary file formats and open standard file formats as well. Okay. So I want to dive into this. I've mentioned kind of image data and metadata already. So I want to think about, when I say Omero Plus is a data management platform, what is the data that is under management in Omero Plus? So it's images, okay? So image files are something that Omero Plus knows where those image files are, and of course how to read them through bioformats. But it's not just the image files. Arguably, I can have a really nice organization on my file system of all my acquired microscope files, and I can name the folders appropriately with dates, and that's data management in a way. So it's not just the files themselves that are under management in Omero Plus. We also have search indices, which allow you to find the data that you care about, in particular, sometimes historical data that you want to see, hey, has someone acquired this data before? Or I know that someone did, where is it? And actually find that at a later date. And metadata. So that metadata could be metadata from the original file. So there's lots of really essential metadata that in those proprietary file formats describes, the laser power, the stage position, maybe the temperature, various things about that acquisition. But there's other metadata that may not exist in that file. Maybe it's in an electronic lab notebook or a LIMS system. Maybe it's written down on a paper towel. And so all of that metadata is information that you can get, that you can bring in to Omero Plus, tagging those images with compound information, maybe patient information, anything that describes the experiment. And of course, that goes back into those search indices and your ability to find the data that you care about. Analytical data is also metadata. We sometimes think of it differently because it is often quite high volume, but analytical metadata can also live alongside the images. And this can be quite large. So let's say you've segmented an image, or things like spatial transcriptomics, we've heard a lot about that across this meeting, that kind of tabular data that's associated with an image acquisition can be included in the Omero Plus platform. And so we have many groups that are expanding beyond the image. Certainly it's a very image-centric platform, but you can see that there's lots that surrounds that image and enriches the management and the awareness of what that image data represents. But all of that is running on a server somewhere. And you as the end user, or your users, are interacting with this data remotely. And we find that actually quite powerful, because it means that I can be anywhere. I don't need to be on that workstation that has the particular software installed and maybe has a license associated with it. I can be at this meeting and I can search for my image data or look at my image data. And so how might you be interacting with your image data remotely? I would say it's probably in one of these three categories. So you might be acquiring new data, you're producing the data, and you're actually getting it into the system, the data management system. You could be looking at data, sharing data, annotating data, even analyzing data from an interface that is often browser-based. So again, this goes back to being able to do it from anywhere. If all the interfaces are in the browser, I don't need a very powerful workstation to look at my data, to interact with my data, and I also don't need a particular software installed. We test with all the major browsers that are popular amongst our users. And so that's the major way that you're interacting with the image data. Now, you may be programmatically inclined, especially in a research computing session. So we also have a number of open APIs in things like Python and MATLAB and Java. And so if you want to write a custom image analysis script or a post-processing script, or even something that's generating a report. It may not be a very compute-heavy workflow, but just something that's quite custom. It's quite easy to build on top of this platform. And so in general, this entire platform, remember that open source core, that's really essential to the way that we think about this software. And so the ability to create your own scripts, to even create your own browser-based web apps, or to customize the metadata that you care about. There's lots of things that are available out of the box, but the platform is purposely very flexible so that you can do what you want to do with it. And so just to give you some examples of this interoperability, there's a number of existing integrations that we have with Omero Plus because we're not necessarily trying to be the analysis platform or even be the visualization platform. We know that there won't be one visualization tool that works across all these domains, or certainly not one analysis tool that will work across these domains. And so what we do a lot of is integrating with the industry standards and integrating with with the platforms that our customers say these are the essential pieces. And so, of course, there's a lot of things from the Python toolbox or R toolbox up there on that screen, MATLAB as well, but other popular tools like Fiji or Cell Profiler. We've seen a lot of those in talks at this meeting. QPath, another great open source analysis option. But also commercial things like Halo and Visiopharm. We have integrations with all of these platforms. Again, because we're not trying to prescribe what you do with your imaging data. We're often talking about we're building the pipes to make all the things that you do with your imaging data easier and a bit more scalable. Okay, so just another brief diagram to say how all these pieces are working together. So you have all your image data is on some file storage. And remember, this is in your infrastructure. So in most cases, you don't want to send all your data to Glencoe software. You have your imaging data on some storage in your institution, and that's likely where it should stay. And remember, it's in lots of different file formats. And so Omero Plus running on a server somewhere, typically in your infrastructures, that could be an on-prem data center. It could be in the cloud, but it could be your cloud environment. And Omero Plus is running on that server, and it's using bioformats to read from all those various proprietary file formats. We have a database, and the images are not going directly into that database, but things like metadata, regions of interest, analytics, that's what's going into the database. But your users are interacting with any of these interfaces at the top. I'm not going to go into all these interfaces in detail, but I just wanted to name some of them. If any of them sound interesting, happy to have a more detailed conversation. But remember, this is all about the sort of analyze, share, collaborate, all those activities that we've described since the beginning. So PathViewer and PathViewer Grid are digital pathology focused interfaces, tools for viewing and annotating large whole slide images. Pageant is a data mining tool, so things like spatial transcriptomics or single cell segmentation or any kind of segmentation. If you want to mine through those results, that's what Pageant is for. We have a number of analysis integrations. I've already named a few. One is called the segmentation connector. This is where you can actually execute your analysis via the browser, and we integrate with the compute resources that your institution cares about on the back end. And then, of course, you can build your own custom pipelines and apps with the open APIs that I've described. Okay, so I next want to just finish with an example of a new direction that we're taking based on some feedback that we've gotten at meetings like this at ABRF in building a bridge between open source Omero and Omero Plus. So we consider this new product or this new tier of Omero Plus called Omero Plus Essentials as a step towards democratizing data management solutions. And I'll tell you why. So today, there are two major camps of options if you want to get started with image data management with Omero. So there's open source Omero. It's freely available. You could get started with it today. If your institution is happy to take on the burden of installing the platform, maintaining it, training users, onboarding users, troubleshooting issues. It's a great open source community, and actually there are many very successful cases of this working incredibly well. But it does require your institution, your staff to take on that burden, that personnel burden, and also the infrastructure burden. In contrast, you could work with Omero Plus, you could work with Glencoe software. In large part, we offset the personnel burden. We install the software, we support it. We can also even train your users. We can meet with them on a regular basis to understand how their imaging needs are evolving. And so we offset the internal staffing requirements effectively to run a data management solution. And that's why people often work with Omero Plus and with Glencoe software when the scale of these solutions becomes much larger, when it becomes beyond a single person's ability to sort of day-to-day work with in their role. But in the middle, what we find is that there's often groups who say, we're willing to take this on. We're willing to manage the data management solution. We're willing to meet with our users. We're a core facility, and that's what we do. But we just need some help getting started. Maybe we don't have all the technical expertise from a systems perspective to get this data management solution up and running. And so we'd really like this middle ground where we'll take on the support and we'll work with our users, but we just need your help setting it up. And we don't want this barrier to entry, in particular from a cost perspective. And so Omero Plus Essentials is is aiming to maintain all the good things about Omero and Omero Plus, this vendor and domain agnostic, this sort of flexible solution, this interoperable solution, but it is cloud native and self-service, leveraging infrastructure as code. And so you can stand up an Omero Plus installation in the browser, clicking around and choosing some options, and then our tooling using infrastructure as code gets this server up and running for you. And so we think that this could be quite nice because there's no upfront cost. We'll do it via a monthly subscription rather than an initial upfront cost. And there's no on-site resources needed, and I want to clarify, I mean on-site hardware resources needed. This would be a cloud-native deployment. Of course, there's always personnel needed to run these things. And so this is a step in a different direction. I started with how important service and support is to Glencoe Software, and we have these very close relationships with our customers, but we recognize that sometimes you're a small group and you don't want that. You don't want to spend so much time with us and have this dedicated support engagement with Glencoe Software. And so this, we're hoping, is a little bit of a middle ground. It's a little bit more supported than open source Omero. We'll get things up and running for you, and then we'll basically hand it over to you, and then you run it from thereafter. But it's a much lower barrier to entry, and it's more of a recurrent cost than something that's upfront. So we're really interested to hear if this would be, compelling as a model to any of your core facilities. Also happy to answer questions. I'm not diving into the details of how we're doing this, but happy to have those discussions after the talk. And so I'll say thank you so much for listening to hear about these solutions. We're at booth 512, and so if you want to stop by before the end of the day, happy to continue the conversation. Thank you.
Quick questions? Yay, open source software? Yay, open formats?
Yeah. Yeah.
Very nice talk. Do you have any pipelines already being used that are specific for structural biology, or like some part of the cryo-EM, NMR, tomography?
Yeah.
Maybe for me as a core director who's using these, it would be helpful to kind of see the interface to get a feel for it.
Yeah, no, it's a really good question. And in complete honesty, the majority of groups that we're working with are more on the digital pathology, high content screening, fluorescence microscopy domains. This is a domain that actually the person sitting right in front of you is a team member at Glencoe Software, material science expert, and we're trying to expand our offerings in those domains. From our perspective, we always say it takes the right partner because we don't often prescribe the workflows. We want to take your workflows that you say, these are the domain standards, and integrate them with the platform. So would be happy to talk more about what we need to do to demonstrate that we could support that domain appropriately. Yeah.
So back to Omero Plus Essentials.
Yeah.
It's great, you can set it up and you'll hand it over. There's always an operating and maintenance
Yeah.
burden
Yeah.
and expertise
Yes.
that you need, which you're not getting if you're just clicking through how to set this up. And
Exactly.
So have you thought through that and come across that problem?
Yeah, so I think the best evidence for this is that open source Omero is incredibly successful. And there's really great, all the documentation for Omero and Omero Plus is also open. And so, things like forums to discuss issues and documentation to describe, common troubleshooting paths. There are actually pretty large institutions that run open source Omero very successfully. And sometimes it's the barrier to entry to just get the thing up and running and get people to use it. So I would say, on the one hand, I see this as an easier entry to the same kind of support model as open source Omero, that there is great community support for these platforms. On the other side, I think it's a question of scale. So if you want to convince your institution, hey, we need Omero Plus, not Essentials, but the traditional enterprise engagement with Glencoe Software, how do you prove to them that it's worth it from a budget perspective? One path that we've seen groups use is they stand up an open source Omero, they get some data imported, they get some users excited, and then they can demonstrate, look, the users want this, and they will use it if we just make it available to them. And so we think that Omero Plus Essentials could be an easier entry to get something stood up, get some data imported, and then you can within your organization say, look, we were able to run it, we were able to get some data imported and the users want it, and it's potentially a path that you don't run Omero Plus Essentials for 10 years, maybe you run it for 1 or 2 years, and then you ultimately transition. Once you have 10 cores excited about it, then you transition to more of the enterprise version. So I see it as a third step. So there's always been open source Omero and Omero Plus, and maybe this is the middle ground, but it's part of a progression that we've seen groups travel successfully before. Yeah, of course. Thank you so much.
Thank you very much, and thank you to Glencoe for sponsoring this session.
Absolutely.
All right, great. So I'm Jason Key. I'm the Associate Director of Technology at SBGrid, Harvard Medical School in Boston. I'm also a lecturer in the Department of Biological Chemistry and Molecular Pharmacology. I'm a structural biologist by background, and I found myself drifting more into the computational sides of things, and before you knew it, I was racking servers and putting disks in file servers and GPUs, and I found my home at SBGrid, which is a sort of a research computing support team for structural biologists based in the sort of structural biology community at Harvard. And when they reached out to me to host this meeting, I thought, well, this is really great because I can talk about kind of the nuts and bolts, the sort of guts of how we do what we do, because we're sort of unique as a research computing support team because our primary offering is a software stack, like a collected, distributed, curated software offering. So we're distributed. We're not really localized, and we have members all over the world. So I thought it would be interesting to compare and contrast what my core facility does with those of others. I'm going to touch on some of the services we use, some of the things that are great, some of the things that are not so great, and I'd encourage some discussion. I think we've got some time, and I don't mind being interrupted, so we're not a big group. Feel free to interrupt, raise your hand. So our fundamental goal as a research computing support team is to minimize the barrier between data and results, right? So researchers collect their data, there's a bunch of computation. That computation is maybe not really interesting or even related to the scientific results that they wish to derive from their data, but it's a huge barrier, and sometimes it's insurmountable. Sometimes, of course, people need to go and learn how to write code, or they need to do more work to figure out, like, computationally what, but sometimes it's just software's hard to use, software's written by scientists, software runs on operating systems that are derived in the 50s at AT&T or something, right? These Unix-based high-performance computing, they're not necessarily the sort of intuitive environments for processing data. So what we do is we try to help researchers get over that barrier and get their papers out, right? So structural biology has always been at the forefront of computing. So I put a few things up here on the slide. So this is UCSF in 1965. That's the sort of first visualization, some of the first visualization of protein models in computer. We sort of moved over to these SGI-based systems. They ran IRIX, still a Unix system. Believe it or not, these things cost like $20,000, $25,000 just for your like desktop workstation, right? So structural biology, you could always tell a structural biology lab because you'd go and there would be a dark room in the back and it would be full of these machines, and there would be people in there like looking at, with their goggles on or their 3D vision, trying to trace density or build models. And there was usually somebody who had to maintain all this stuff, and it usually wasn't their job, right? So there was some postdoc or someone in the lab, sort of research associate. For a while, that was me at the University of Chicago. And so we maintain all these things, and it's always been Unix. It's always been pushing the boundaries. And so this is what our sort of modern workstation, I guess, looks like today. This is our SBGrid data center. So we currently have petabytes of storage. I think we counted up 400 GPUs. We had 401 last I checked, so we went over the 400 mark. So the computing just keeps going further and further, and we're just trying to keep up now, mostly with cryo-EM. So who we are. So my group is made up of we're a nonprofit group. We're based at Harvard Medical School, and we're made up of a diverse team of structural biologists like myself, Pete Meyer, Sean Rawson, but we also have a team of system administrators and IT pros. We have software engineers, a couple of programmers. We have a cloud architect. We have occasional postdocs and students. And our goal, again, is just to shepherd structural biologists through their sort of data journey to solving structures. So how do we define structural biology? Because I know that maybe not everybody here is a structural biologist. So for data derived from X-ray crystallography, cryo-electron microscopy and tomography, that's kind of the main, I think, the main data volume these days. NMR, of course, is a popular technique mostly for looking at in drug discovery these days, for looking at compound binding fragments, protein dynamics. Molecular dynamics is computationally looking at those molecular dynamics. Drug discovery by virtual screening and computational chemistry. We count that within structural biology as it's, so you're using molecular models to make conclusions about what would bind or what wouldn't bind. And this is kind of the next big thing by sort of hot software titles is structural prediction and modeling, right? So this started with Alphafold, Fold, and these tools just keep coming. So this is from 2 days ago. This is a generative AI. I mean, this is the New York Times, right? So the New York Times comes up and there's molecular models of CRISPR, and so this is a company. I think they're called Profluent or something. And they've built an AI model to build CRISPR structures, right? This is it's open, they say. It's in GitHub. I went to their GitHub. There's no code there. It's just a protein sequence and a transfection protocol. but it's conceivably open. But these tools just keep coming, right? And they use a sort of similar set of architecture to cryo-EM. They need GPUs. They need a lot of storage. They need performant computing. So we're adding these all the time, and the whole goal here is understanding molecular mechanism at atomic scale. So that's what we do. That's what we want to help researchers do. So how do we do that? We have hardware. So we have, this is our sort of primary data center. We've got a couple of others scattered around. We've got computing and storage, and our primary role is to support the cryo-EM facility at HMS. But we also have computing in here for these AI-based approaches for sort of docking and proteomics and protein folding. We also support a lot of workstations and Linux and Mac around HMS in the Boston area. So in that way, we're sort of like a conventional research computing team. We also do training, so we have workshops. This is one we did a couple of weeks ago. This is our, we call the Train the Trainer workshop, where we're bringing in PIs from all over, and we're training them on certain certain structural biology tools with the expectation that they will take those materials and what they learn back to their lab and train their staff. So, and this is really focused on minority-serving institutions and historically black colleges, but it's really open to everybody. But the first 2 rounds we've brought in, this is Brian, he's from Meharry Medical College in Nashville. So we've brought in staff from other universities, hosted these at HMS, and we're going to ultimately move in to rely on another cryo-EM tools. We have a webinar series. You can find those on YouTube. We have hundreds of structural biology talks recorded on, and you can listen to them. And our most recent, I think we have a Rely-On one here. Now this is RF diffusion. There's a Rely-On one. We host data, so we have a diffraction data database. I'm not going to talk about this too much today, but if you go to the PDB and you deposit coordinates from a structure, if it's an X-ray diffraction structure, you can put your raw data into our database, and it will be linked in the protein data bank. And then this is the main thing. This is, I think, what most people know as far as this scientific software stack. Right? So we actively curate and install and maintain 500 plus scientific applications for structural biology. And within those applications, we have multiple versions of each. So in the end, it ends up being thousands of different versions of software. These are curated by structural biologists, tested for Mac OS and Linux, and you can see the whole list at sbgrid.org. So SBGrid is growing. It's more than 520 labs now. You can see the distribution here. We don't have anyone in Africa yet, but we did have an inquiry from South Africa, so maybe that's the last continent. We have one lone lab in Montevideo, Uruguay, but otherwise we're sort of heavily represented in the Boston area and Europe, but we have labs all over the world using the software. They're academic, they're government, they're commercial. Some of the biggest pharma companies, some of the smallest pharma companies. There are pharma companies that have one structural biologist who use SBGrid. We have estimates of about 2,000 unique users per month, so we have a lot of users. And the software is used in cloud, HPC, workstation, laptops, anywhere that you can run Linux, Mac OS, you can run SBGrid. So because of the size here, as a core, scalability is essential to everything that we do. So as I go through and talk about some of the services we use, we're always thinking, how do we scale this? How can we make this? How can we do this without a one-off time commitment so that we can make this work for 500 labs, right? So I'm going to talk about curating the software and managing the software environments, some of the tools we use, how we do it, the software packaging and distribution, what we built, what we bought, services that we use through cloud and HMS. I'd like to talk a little bit about the community engagement, because that's an important part of what we do. And then if I have some time, I'll talk about some new stuff we're building to give people access to compute resources. So how are we doing? All right, we're good. All right. So software curation, where does all this software come from? We don't write this software. Right? So this is structural biology software almost entirely written by scientist developers who are working in academia. Some have big grant-funded projects that pay for their developers, CCP4, Phoenix. I guess these are the sort of big crystallography ones that come to mind. Then we also have scientists who are working in academia for their own research projects who are writing software to solve their problems, publishing it, making it available, but it's not necessarily getting paid for out of their funding. They just need it to do their work. We also have a number of titles from private companies. So Schrodinger, Global Phasing makes a popular X-ray crystallography data processing suite used in pharma. And then tons of open source projects from academia and industry. Just a couple of examples. We've got this new tool, Chroma, which came out of a group in Boston, this pharma called Generate that's using AI tools to define protein structure based on their own, I guess, reverse polymer diffusion algorithms using AI. And they can do interesting things where they can describe a protein in language and have their model build it. They built models of every letter of the alphabet. They can sort of codify in melting temps. And so we'll see if this is borne out in actual, like, real proteins. But that software is open source, and we have it available. And of course, Alphafold from Google's DeepMind is an open source title written by a gigantic company. So we have many different types of software, license types. Many are not open. Some have to be restricted. So we have to manage all of this. So this is what the workflow looks like. We sort of build and test the software. We make the environment. We package it up. We have to include all the dependency system libraries and metadata. We actually do the distribution. And then we're building some tools to give access to compute resources that I'll talk about here at the end. I just want to note that we actually have two collections of software. We have SBGrid, but we have another collection called BioGrids. We have a team of bioinformatics pros who build and install software from bioinformatics. I don't know what most of these things do. A lot of them end in seek, and I'm lucky that they do. It's primarily in use at Boston Children's Hospital and HMS for processing bioinformatics data. But when you add up everything that we're distributing, we're currently supporting more than 5 terabytes of software. Here's what the growth in that software stack looks like. So if you start here, it's kind of hard to see. This is 2010, and this is the number of titles in the collection. And this is the number of versions. So We started out in the 2,000 versions or something. We're up past 5,000 now. And so a number of titles were up past 500. And it just keeps going. I take some comfort in the fact that it's kind of been over, I guess, if you look at it. But I don't know. There's more and more cryo-EM and AI software coming all the time. So, so this is what building scientific software is like, right? So we have the scientific applications up here. This is a popular cartoon from the XKCD comic. The scientific applications themselves are relatively small. They usually don't have a lot to them, but they sort of sit on a lot of dependencies. So these do the math, I/O, graphics network, all of the important things that sort of computing rely on this sort of core set of application libraries. So, and then there's these OS level components. So we install this part, right? So we install as much of this as we can to make the software portable, robust, and reliable across multiple operating systems. So to do that, we have a whole infrastructure of machines. So we've got dedicated build machines for Mac OS and Linux. We keep virtual machines and we keep hardware machines. This is just a console from our virtual machine platform. This is the upstream open source product from Red Hat. They make it available. It's called Overt. This is great because it allows us to just bring up machines whenever we need them. We can arbitrarily define the CPU types. We can build for certain CPUs. We can test for certain CPUs. It doesn't do GPUs well. That's why we keep some like real actual hardware around. We build on the oldest supporting operating system so that it's the most compatible. Right now that's CentOS 7, which is ancient. It's 10 years old in June. So it's going to be end of life at the end of June, and we'll have to move on to Rocky 8. But it will be, which is also still pretty old. But that gives us the best compatibility across operating systems. We impose the minimal requirements. I showed you that diagram. We try to include all of those libraries with each build. That does sacrifice some hard drive space, but it gives us the most sustainable and installable stack of software. It also sort of conserves the blast radius when something's broken or has a bug or something is not working. We isolate everything within HMS, so all of our curators, we have our own VLAN. SBGrid within HMS, we have our own firewall, our own network, we have our own VLANs, so all of the software curation sits on its own independent network. No users log in over there, no users have access to that network, and that's done just mostly with security in mind, but it also allows us to build in specialized logging. So everything that every one of our curators does is logged. So if I want to know what somebody did when they installed software in 2014, I can go find out. Like it's all, every command ever run for the last decade is in our logs, right? So it's also really handy when you try to figure out how was this installed 10 years ago, right? So you can go and look it up. We share all the tool chains, and they have to be approved by me, basically. So they're all, every compiler that we install, every system that we use to build software, it's shared among the group so that we don't have a bunch of people doing different things, because that's when problems come up. That's when inconsistencies arise. We want everything to be standardized on the same set of tools at all times. And critically, we use ZFS as a file system, which sounds like an arcane thing, but ZFS is a phenomenally amazing tool. It allows snapshots, like granular snapshots. So we snapshot every 3 minutes. And so if there's a mistake or something happens, we can always roll back. So it allows the curators to go in, run software, test software. You wouldn't believe how often there's a script or something that has an RM minus, /* or something in it, right? That somebody wrote. They bundled it in, and they're like, "Oops, I accidentally did something bad." We can roll it all back. It's all right. How do we test? So we test on Red Hat derivatives. So we kind of give best effort for other Linux distributions like Ubuntu, but we have tons of Ubuntu and Debian users. So we hear about bugs pretty fast, and we try to fix them where we can. I don't know of any that we haven't been able to sort of fix or get around. We also have the same isolated network and test accounts. We have a defined testing workflow, so we have a chart. Every curator has to go through, fill out the chart for every title. So we can go and say, when we get a ticket in 6 months, like, why is this broken? Oh, it was broken when it went out. You just, you marked it in the sheet, and then we can figure out how to fix it. We automate as much of this as we can, and I'll show you more of that in a second. And we don't support Windows yet, but there is some ability to support Linux in the Windows subsystem for Linux. We just haven't dedicated resources to that, but I think it could be really useful. As soon as the GPU support there is good and we can sort of verify that, we'd be able to reach a whole new community of structural biology users. I'm just showing here all of our test machines. They have little thumbnails. In our framework, you can go and click on any of these. They come up in your browser, and you can like run and test. Within the SBGrid network. So what are users running? So most of our users run. It's dominated by Linux, right? So almost all of the software is run on Linux. Alma is really the most popular right now. This is sort of troubling because it only has about 6 weeks of life left, but it is shrinking. Rocky is making an appearance, and then we've got some bit players over here, Ubuntu and Debian. Right? But if you look at things like a graphical application, if you look at like PyMol, right? So Mac is really prevalent, right? Like we see most people are doing graphics and visualization on Macs. Still a lot of CentOS 7, but so we still need to make the case like Macs need to be supported. As problematic as they are with the new operating systems, the new CPUs, the new security restraints, package signing, people are still using them, and we intend to keep supporting them as much as we can. So just to detail some of the tools we use here, as much open source as possible. Internally, we use an environment module system to manage compiler variables, which is pretty handy. We use some package installers, Combo, Mamba, Pixie, so these are all different versions of the same thing. Pixie is the newest, fastest version of Conda. And there's a tool called SPAC, which is great for building packages for high-performance computing. We use these where applicable. We follow the instructions that the developers give for installing their software, and often they say to use these tools, so that's what we do. I showed you the Real VNC dashboard on the previous page. We containerize some things. This is kind of you don't really need to containerize SBGrid because we work really hard to make it run on bare metal, but still there's some real benefits to containerization. So we do use those tools internally and are working on some docs to sort of roll that out a little more broadly. And GitLab is huge. GitLab is our own self-hosted. It's like GitHub or Bitbucket, but it's a self-hosted product, and it's fantastic and has become a huge part of everything we do. I'm showing it here. What I'm showing is these are the software development team. They're committing software and committing the changes as they go, and it's running automated tests and procedures on all of it. Everybody can see everything that everybody's working on. We can see, okay, this is successful or this is failing. We use it to manage our workflow. We manage our issues. We keep our code there. It's a beautiful, amazing open source thing. So here's another view of our GitLab pipeline. When we add a new title, so I added CS2 star, I think this was like 2 weeks ago. We run through and build all the different environments. We do some sanity testing on different platforms. And then we build our SBGrid software environment. It's all automated within the process. We scan for viruses. We fix all the permissions. We make all the symlinks relative. We build module files in case people want to run this in LMOD or in some HPC facility. And we build our own sort of SBGrid environment within. I'm going to talk about that just for a second now. This needs like a quick primer. Just for those of you who are not Unix nerds, all software that's run in a Unix environment inherits what's known as a shell environment. So in the environment, there's a bunch of variables that are specified. When an application runs, it inherits those, and it changes the way that that application works. And you might remember, if you've ever reset your path variable to something wrong, and all your applications disappeared, that's because that variable defines where everything is, right? So there's a lot of variables that are sort of built in and are standard to the shell, but applications can define them arbitrarily, just as the developer sees fit, right? And so I've listed some of the ones here that you might be familiar with. This all has to be managed per application. If you, say, have used software where you have to source in a file, or if you have to load a config, or maybe module load something, what you're doing is setting all these variables in your environment. Right? So we wanted to make this go away. Like, we wanted to make this part of the background for users do not have to care how any of this stuff works. So we built a system. Oh, well, first I just want to show how complicated this gets. I forgot that this is CCP4. It's a suite for X-ray crystallography and also some cryo-EM tools. And so this just shows all of the variables that have to be set in your shell just for this to work properly, right? This is, I'm not even sure this is all of them. And some of these are specific to CCP4, like it sets a web browser, which I guess makes sense. It could just call whichever one was in the path, but it needs to have a variable. But it also sets like where its SSL certificates come from. So normally those would come from your operating system, but CCP4 needs to have its own. So this is a lot, right? So users have to manage all of this stuff for all of their software titles. They're often mutually exclusive, so you can't have one software title loaded while you have another software title loaded. So to make this simpler, we built a system called capsules, where we write up all of this environment into a description file, and then we generate an executable off of that file with the same name as what you'd want to call. And when you call that, it builds out the whole environment, runs your application, and then the environment's gone when the application exits. So this way, users don't have to know what gets set or what needs to be set, what applications share variables, what don't. We do all of that behind the scenes. Just a quick, this is in just a figure executable. It opens a file descriptor, so it's a Unix term. It opens its file, it allocates its memory. It loads that environment, it runs, right? So in capsules, the executable shell is here. This is your terminal where you're working. It defines this environment, and then it loads this environment, which might have some things in common with what you have in your shell, but we set it to be defined so that anything that would break the application or interfere with its interaction with other applications, those things get unset. So, and we can make them available to the user so that they could set them if they wanted, but for the most part, it should just work. And so this is great because in a sort of standard shell, if you have a workflow where you have one executable that calls a second one, they have to live in this same space. But this isn't always possible. So in capsules, it works like this, where you call it, this environment is defined, then it calls this one, and this environment is defined. And we can do really complicated things like have multiple Python environments, have multiple R environments, have Python environments that call different mutually exclusive sets of libraries that are linked using LD library path or other shell variables to load CUDA or other, we can unload CUDA so that local versions of CUDA that would break your install wouldn't break your software. So it's a nice way to give every executable its own environment and make it all disappear behind the scenes. A couple of nice things is everything works unchanged. You don't have to load anything. You don't have to script anything. You just run it just like the developer has in their tutorial, like on their wiki. No user management, right? Everything's predictable, so we know what runs what. We know what can see what. We know what executables can call other executables. So it turns out that if you have 500 applications installed in a software tree, you have duplicates, right? Because you can only name executables so many things. There's, you know, refine as a name for structural biology executable has been used more than one time. Analyze, FFT, like it turns out that that gets used a lot. So you may not want the refine from EMAN2 when you're working in global phasing, right? So you're processing X-ray data. You don't want the cryo-EM version of refine. So in the SBGrid stack, this is all defined and it's not left to chance. You'll get the one that you intend to get. We can also manage versions, so you can set the version that you want to run on the fly. And you can also log all this, and you can see everything that you're doing. Right? So it makes it much cleaner, easier. And frankly, without this, we wouldn't be able to install this much software into one space. It just wouldn't be possible. So one quick example, this is Coot. Coot has an ATP in its beak. This is the figure I showed you at the beginning, my FAD. So we can use capsules to impose arguments on programs that don't have them, right? So we can put SBGrid arguments onto them and then give users functionality to manage their software on the fly. So here it's just running the SBApp help message, which tells you like all the different arguments you can give it. So just a quick example, you can say coot SBApp:L. It lists every title in SBGrid that has coot in it, which is handy to know because there's 3 different coots in 3 different titles, right? So this extends to lots of different arguments. We use this for obsoleting titles. We use this to put in messages when a license file is missing that a user needs to add. It allows us to communicate directly with the user from the command line. We do have to be careful to make sure we preface our message with like, this is a message from SBGrid, not coot, right? And we tried to make that very clear. But it's super powerful, and we use it to make managing software sane. So we also use it so that in HPC environments, you can load everything into LMOD. So I don't know if you've used modules in LMOD. You do like a module load. It's seamless to integrate this with LMOD. So, and the reason we did this is because people use that interface in HPC. They're used to that interface. They don't want a new interface. I don't want to have to source SBGrid. I just want to use my modules. Well, here it is. It's all there. It's all ready to go in modules. Back to the testing, we can use the capsules to run any test we want for any executable we want, and we can do it all in GitLab every time we commit. So we have executable testing for every title, and we run it on every distro and Mac that we've got, right? So this is great because we can set up test data, we can run help version, we can see all of the dependencies, and we can just see what works, what doesn't work on the fly, all automated. These tests kind of look like this. If you zoom in, you can see it's just running just different commands. This one's High-C Explorer. It's from yesterday. And so it's just running through and running -- version for all of the binaries in High-C Explorer, which is a very simple test, but you'd be surprised how many times this fails. If it's missing a library, if it's missing an environment variable, this will fail. It will flag it, and we'll see High-C Explorer is broken on Ubuntu. But, oh, and I just want to stress right here that since we're doing this for all versions on all platforms, if we make a change, like Apple releases an operating system, or we try to support Amazon Linux 3 or whatever it is, we can run our tests and we can see how does all this pan out, and we can also see when new OSs come out, like what's broken that's been installed previously. We don't usually go back and test software from 5 years ago, right? But we can easily do it because we have all this built in and automated. So this is all documented on our website. You can check it all out. Let's see. So a question I get asked a lot is how do capsules compare to containers? So they're much lighter. They're just subshells. You're essentially programmatically wrapping everything, but they're compatible with all platforms and all containers. So you can put capsules in containers. It all works. Containers have flexibility with namespaces. That is, you can just redefine your paths. You can't do that in capsules. And we don't have any packaging or repository for capsules. Like containers, you can do like a Docker pull, and it'll pull in your image. Or Singularity has this as well. We don't have any of that built in. We do that using different tools. So, all right, I thought that was a lot all at one time. I'm going to sort of shift a little bit. I thought maybe a break just to the wall, just no more Unix stuff. The rest is like pretty infrastructure. Any questions on any of that? All right. All right, cool. All right. So how do we get software out to people? So we looked at a couple of different ways to do this. There are package managers out there. You can use operating systems that have RPMs. There's tools like Brew and Conda. None of them really suited our needs. mostly because we'd already built a whole infrastructure for distributing software, and we would have to change all of that to shoehorn it into a package manager, right? So we just built our own. And we built 2 versions of that. We've got a graphical version. I've heard it referred to sort of like an app store. You don't have to pay for anything. It's all there. You can just install it. But it is App Store-like in that it's kind of a GUI, and you can click it when the software gets installed. It's available for Mac and Linux. And then we have a command line tool with just showing you this sort of terminal output. This is great because it allows you to install software remotely over SSH in the cloud or in containers. You can also script everything up in here, and it's sort of my tool of choice. Just talking about the tools we use, the graphics here is an Electron app. So if you've ever used Spotify or Slack, they're also Electron. They're a web browser, a stripped down version of Chrome called chromium that's running essentially web code, right? And ours is running Angular, TypeScript. Angular is TypeScript. It was developed by Google. It's essentially like a less Wild West version of JavaScript. The command line is also Angular and TypeScript, and we build and test all of these in GitLab. So we have automated tests for all of these things. This was a good choice for us because we already had all of the software going to the web. We had APIs already. This is just a browser that can load all this data like it was loading it from our web page with a few simple APIs. So it allowed us to really seamlessly jump in and make this installation manager work. And that's because the metadata is complicated, right? So managing software includes a lot of different metadata, and we struggle to maintain all of this stuff. So you need title, versions, the websites, the developers' names, the license, the distributions, OSs, our relevant MOUs with all the developers, the webinars we've done, the date we've installed it, any usage info for users. We keep the publications, all of the dependencies, the other titles they rely on. We also use it to manage user accounts so they can access it, authentication for those user accounts. They're all individually licensed, so they all need to have that. We have stories about our developers that go on our website. We keep publications for all of our members. We invoice people. We asset tag. I'm sure there are more things. All of this has to go from our database out to our installation manager with our APIs. It goes to our websites. We bill and invoice people for SBGrid membership. And then we have mailing lists. We have to correspond with people. We have web forms where people fill out to access to get accounts and such. So how do we deal with all this? So we ended up building our own distribution infrastructure dashboard. We did this in Django, which is a Python framework for web, and it allowed us to just build out all the installation profiles for all our users and do logic to calculate who should get what software and who shouldn't have access to other software titles. We keep all the software title versions of metadata, we authenticate users, and then all this has APIs that go out to the web and the installation manager. We automate everything we can here. So everything, we try not to ever have to enter anything manually. Just a quick view, this is a software title. It has just all of the available versions, all the OSs, the size, which versions we sort of send by default, when we added it. And so all of this is managed internally for every title. We have every version, every title, thousands of titles, and it's all automated from our deployment, right? So everything that gets added here is done through APIs, through some Python. So most importantly, we manage, our members come from certain groups, academic, beamline, government, industry. Each group has access to a different set of licenses for title. If you're an academic, you get access to the academic-only software. If you're industry, you only get open source software, unless you have licenses. And then government and beamlines are usually somewhere in between. So some developers draw a line at government and say, no licenses for government. Some say, government is nonprofit, so we have built logic in that depending on who you are, you have access to a certain set of titles that you would be licensed to use. And then we can also classify by group, because sometimes people say, I don't do NMR, I don't want any of the NMR software, so we can take it out of their profile. We can add in just by title, so I'm just showing just a bunch of different titles that we've added in. This is just my reference testing site where I add in all the titles. So we have custom configurations for each installation profile, and then each lab is within that installation profile. So everyone in that lab gets the same licenses. So that way, since the licenses are defined at the PI level, everybody has access to all the same software, all within their own license group. It's very easy and convenient once we have it configured and set up, and we don't have to worry about people getting the wrong things. All right, but we outgrew this thing, right? So, and we outgrew it because it didn't do a good job of handling people. Like, we needed to keep track of our members, we needed to keep track of our users, but not just users, we needed developers, technical staff, administrative contacts. Like, these aren't people who are using the software, but they're relevant to everything that we do. So we had to either keep continually adding more functionality to this thing, which we didn't want to do because it's a constant burden, and every line of code you write, you have to maintain, right? Or we could go buy something, right, and try to use something else. And if we did, maybe we wouldn't have to host it, because we host enough stuff. So it'd be nice to have a thing that was hosted by someone else, and when it goes down, we don't have to run around and try and bring it back up, right? Hopefully it doesn't go down. Hopefully it's backed up, right? So also, can it be administrator friendly, right? So half of the team are Unix nerds and structural biologists who are comfortable getting into the guts of these things, but our administrators would like to be able to add new fields and change functionality without having to learn Python, right? So, so we looked around. Ultimately, we landed on a CRM. So we tried a few. We tried one called Sugar, which is open source. We tried a handful. We ultimately landed on Salesforce. It has a terrible name. Because we don't really use it to sell anything. We just use it to manage all of the metadata around software titles and users and groups and send email. And that's really it. We integrated it with our Django-based infrastructure, so it syncs back to everything we have in Django, so all of our APIs that we wrote in Python all work. It's not what we wanted. Like, it requires constant maintenance. So we have a whole team of people who have to develop this thing. Like, half of it didn't work out of the box. Like, it's probably better than what we would have built, but, and it can be extended for a lot of other uses, but they all need money, right? So doing it for free or doing it on the cheap, we have an academic license, so it's actually pretty cheap compared to like a regular Salesforce deployment. But if I had to do it over again, I don't know that I would do it that way. But the administrators seem more or less happy now that things are stood up and they're working. So for managing your cores, if you're thinking about a CRM, the primary consideration that led to this is, will it be around in 10 years? Like, if we build this thing, can we like depend on it being there? If we pick some startup that has like an amazing product, like, will they still be around? Are we going to inherit some open source thing that we have to fork and maintain ourselves? Or is it going to be like HubSpot and it's going to like track everybody doing everything that they do, and we don't, right? We don't, so this is where we ended up. All right, so how does software get to users from there, right? So we've got our web API, we've got a whole pool of servers that we push software to. We push from our development tree that I showed you from GitLab. It goes here, it gets replicated across these, and there's a load balancer that manages it all, and then users get the metadata that tell them what they have access to and what's available, and then they can transfer it directly to themselves by rsync. And recently we started going through AWS. So we can go through AWS Lambda, which is just a service that you can use to run like simple serverless operations, to make a tarball that we stick in their object storage and then you can pull it through CloudFront directly. So this has some benefits. You get certificates, so you know you're getting what you're getting. The tarballs are signed. So if you download something and it's corrupt, the installation manager will complain. Also, if somebody got access to our repo and put a bunch of corrupt stuff in there, it would show up as corrupt because they can't sign our cert. So it's much more secure, and we don't have to maintain a whole pool of servers to run it. So this is probably the future. So, and certainly is in use right now for a lot of users, because many users can't do rsync because it's becoming less and less available. Certainly within outside of academic institutions, and even in academic institutions, we usually have to get some firewall exceptions just to get it through the firewall. So how do we do user support? We do user support mostly by email. We use a self-hosted system called RT, Request Tracker. It's ancient, it's written in Perl. But it's self-hosted, and we have the database, and we keep it all in-house, and it works really well. So as much as I would like to move away from our request tracker, I think my team would come at me with pitchforks every time I even sort of talk about it. It's like, yeah, I can't. Eventually there'll be some new tool with like AI features or something, and it'll be so compelling I can like make the sale. But right now, RT is where we're staying. All new accounts show up in here, bug fixes, people requesting new titles, new versions. And so just showing how this all shows up as tickets. We assign these to people, and then there's some email correspondence. We can all kind of see what's going on. We can comment back and forth to each other, like, hey, is this broken? Hey, it was broken last week, but I thought I fixed it, that sort of thing. And then, keep touch with the user all the while and make sure that these things actually get seen through. And then it has statistics, so we can go and say, like, this has been open a long time. We can pass things around when people are out, right? I go away for a week, somebody might go and see, oh, Jason's tickets are still open, let's make sure there's nothing on them. So it's good, and it's sort of standard in IT. I just want to put on here like Zoom and Slack. So this has been happening lately. One-on-one support via Zoom is good. You solve the problem like almost every time. You can do remote control. You can set up office hours. People can log in. It's not very scalable. Like, it takes a lot of time. So we have to sort of figure out how we find the like middle ground here. Slack is kind of something somewhere in between this chat-based tool. It's just not, these synchronous approaches are hard, right? Because everybody has to get on the same page. So we all have to set a time, and we all have to be ready, and you have to have access to the machine you want to troubleshoot, which if it's a Linux workstation, maybe it's out in the lab, and so you're out there trying to get Zoom to screen share on an Ubuntu machine that you don't have access to. It's not always great, but it could be great. We just have to work on the tools a little more. We also just don't like accessing people's machines because we're not system administrators, so we shouldn't necessarily be configuring things. But if we can see what people are doing, you can often turn a 10 email exchange into 2 minutes on Zoom, which is huge. We try to document as much as we can. We have a wiki. It's all in Git. It's fantastic because you just commit to Git and it just automatically shows up on the wiki. We use a static site generator called Golem. It's fantastic. I like this because it allows us to document fast and keep everything in a big global history, so it's no big deal to go and delete a bunch of junk because you know it's in the Git repo anyway, and it'll always be there if you need it. So it's great. It could be better. We are looking at other solutions, constantly looking at new wikis and other solutions, and I would like to upgrade this soon. But it's good enough for now, but I think we've outgrown it. All right, driving engagement. All right, we're almost done. So this is important because we have a huge community of users. So we want to be engaged with that community. We have to do asynchronous things because some people are overseas. We try to do synchronous things in person. So we have a webinar series. We do these at least once a month. And the idea is to give the software developers a forum where they can talk to an audience technically and talk about their software, right? That's not possible usually in like scientific meetings. You can't go and talk about like algorithms and routines. There's a few conferences for that, but generally speaking, it's not common. So, and even then, most software developers wouldn't get access to those. So we try to give that forum. And it's been well received. We do lots of these, and we just did Rely on 5, and it was great. I think we had about 200 people. We don't only do super popular titles. We sometimes do really niche things for like one thing in NMR that 5 people come to, but it's still important, so we do them. We record and post and share these. We have regular newsletters, so we send these out every month. We've sent them out every month for 10 years. New versions of software, what we're doing, what we're releasing, announcements for workshops and such. We've been doing collaborative workshops where we go to another institution. Usually we partner with a beamline at APS called NECAT and do like X-ray crystallography. And this is where Salesforce really comes in, like, and is sort of critical here. Like, we keep track of like all of the webinars, all of the workshops, who attended, all this, so that we can keep those channels open, and we know who we've communicated with. And by we, I mean the operations team, because I don't do any of that. But for them, it's fantastic. It's really great. They are able to make sure that, because it's embarrassing if you go and ask Shores to do another Reliant webinar, and he's like, you just asked me 8 months ago. You're like, oh, sorry, right? So we need to have all that data visible to everybody on the team. All right, let's see. We're done at, what, 11:45. I'm going to do 2 minutes to show you guys this, and then I'll wrap. Is that, okay, cool. So we're building what we call SB Cloud. This is the access to compute that I was talking about before, and it's really similar to what Aaron was talking about, right? So we're building a structural biology platform in AWS that you can just drive in your web browser. It's backed all with the SBGrid software, and it's 100% infrastructure as code, which means you open an Amazon account, you run this Terraform, which is a framework for building Amazon infrastructure, and it sets up a whole compute cluster for you with a web browser, and it's got open on demand in the front, tells you how much you're spending. You can open terminals and editors. You can open a desktop. It's actually reasonably speedy. All of the compute is on parallel cluster, so it brings up the instances as you need them and then shuts them down. It uses Slurm to do all the scheduling. It has this Amazon FSx Lustre-based file system, which is highly performant on the back end. But then that data seamlessly gets archived to Amazon's S3, which is their object store. So that's the cheap storage. The Lustre is the super fancy luxury storage. So it allows you to get your data off of the expensive storage as soon as you need to, and keep your costs down. We've got CrowdSpark in there. We've got, this is Chimera X. All of the visualizations are actually GPU accelerated, so you can run it in a browser, and it's actually fast. So that's good. It's a little laggy over Wi-Fi, but when you're plugged in, it's great. The latency is kind of a killer. You don't need to, this is all of the guts of the Amazon stuff, which I'm happy to talk about later if anyone is interested. Right, so we're running this at Harvard, BCH, and at some local pharma companies in Boston. We're also using this for this train the trainer that I mentioned earlier. We're building out this Rely on tutorial in this platform. So we're bringing in PIs from historically black colleges and minority serving institutions, and they can learn Rely on in this shared computing environment. We're doing cost optimization. We're working with a group at KEK, which is in Japan. We've done a lot of work to make Amazon cheaper. We're trying to piggyback off Toshio's work. He's done an amazing job, and he's helping us pilot this. You can do multiple clusters, so everybody in your group could have their own compute cluster if you want. And again, this just gets deployed as code. You have an Amazon account, you deploy this thing, and it's ready to go. And so that's still in flight. We're actively working to bring this to fruition. Yeah, I would say it's sort of late beta right now, but it still needs a little bit of polish to make. The data's a little clunky, and that's because Amazon's cloud. is a little weird. It's not a sort of intuitive model, but we're working to make that better. All right, curation of software. We mostly use home-built stuff, mostly open source tools. We bought some stuff. It doesn't necessarily mean it's amazing. And then managing users, developers, and community. Our CRM has been really huge for that. And then I just want to highlight a couple of challenges we face. 520 labs is a lot, so we're working to scale user support. We just added 2 more staff members with some cryo-EM backgrounds, so that's going to help. One of them is one of the developers of a package called Scipion, which is this massive package out of Spain that is like a little mini SBGrid, so it seems like a really natural fit to have somebody who has worked in that environment, so I'm excited. Software size is getting huge. It's just getting bigger all the time, and these machine learning models are not helping anything, so we'll keep doing it, but keeping the size under control is always a structure, or is always a struggle. And security, right, is always a problem, and I wanted to highlight just this recent. So this was a sustained nation-state attack on a core library that's used in everything. It's used in all over the Linux ecosystem. It's used on Macs. It's used on basically any machine that runs Unix, right? And so this compression library had this code that was inserted in by a malicious developer who cultivated a relationship with the maintainer of this package through social engineering and harassment, managed to work their way into that package, insert all of this code, hid this stuff in a binary test file that was supposed to be corrupt data, and then the build system would go to this file, insert it in, recognize when you're running SSH, and then recognize certain SSH keys so that the whole world would have access to every machine that's running SSH. It took years of work. It was insanely clever, and like, it's terrifying, like that this is out there, right? This is going into open source projects, and they caught it. It's great, but they caught it because one guy was watching, and he said, this is using a little bit more CPU than it usually uses, and he was curious enough to investigate, and he investigated more, and he investigated more, and this went all the way down to just some, I don't know, sustained effort to compromise all SSH access to all machines everywhere. So how much of this is still out there? I don't know. I think we have to assume that all scientific software is inherently insecure and compute accordingly. And we're working on ways to do that. All right, just to acknowledge Pete and Sean, other scientists in the group. Piotr's the director of SBGrid. This is our admin team. Michelle is our lead of operations. Software curators are here: Carol, Jim, Eli, Estrella, and Tom. Tom's based in New Zealand, so he's in Otago, so he's leading our effort on that side of the planet. It's good to have somebody who's on the same time zone as far as user support goes. Ben is our cloud architect. So Ben actually was at SBGrid in like 2007. He sort of helped build SBGrid, and then he left, and he went and worked for Amazon, and he worked for some other companies, and then he decided he wanted to come back, and he's building our cloud platform. Yorgos is also helping build the cloud platform. He also built our installation manager, does a lot of our web stuff. Brian was the one teaching in the Chimera X workshop. He's actually an undergraduate who did one of our sessions, got up to speed with Chimera X, and taught a session. He did a good job. And this is our admin team. Phil, Jeff, Justin, and Franco, they admin all our servers, rack servers, power, cable, network, all that stuff. They swap the disks, keep everything running. And that's it. Any questions? How do we do? All right, just a little over. We got a late start, so. All right. Any questions? Sure.
So a lot of scientific software, especially if it's written by a grad student, for those who project gets abandoned. So what do you do about that?
Yeah.
You still maintain every software that you ever started maintaining.
Yeah, there's a lot of that. And I think software loses funding, developers move on. We have a package in the tree that's called XTLView. It's an old package for viewing X-ray crystallography data. And it still works and it still runs. And somebody put in a ticket and said, like, can you send me that? I think you are the only one who still has XTLView. So I emailed the developer and he said, I don't even have that anymore. I don't do structural biology. I run a model train store, right? And it said, so we are trying to keep these things available. If it's not horribly buggy and it still works as advertised, we keep it. We archive everything that we've ever installed. So anytime anyone says, I wrote a paper 6 years ago, I used this, I can't reproduce that, can you give me that version? We have byte for byte versions of everything we've ever done. So we have a huge library of software. We're always removing things as fast as we're adding them, hopefully, to try and keep that size down. But we keep them. So inevitably things age out and they stop working. So once we know it doesn't work on anything, we just pull them. A good example of that is macOS. 10.14 Mojave was the last macOS that supported 32-bit executables. So anything that wasn't maintained, that there wasn't someone around to build a 64-bit version, all that software is gone, right? So we keep one machine that we can run it on just to make sure we can still run it. But we kept an SGI until like 2 years ago, just in case somebody wanted to like run a thing. But eventually it became a doorstop and somebody said, get rid of this, so. Yeah.
You said request tracker, is that the best practical?
Yes.
Does that tie into GitLab?
Not currently. We would like to have it tied into more of our systems, but right now it's, yeah, it's separate. GitLab's better. It has better request tracking and issues, and ultimately I'd like to move everything over there and have everything be issue-based. There's a service desk functionality there that we could use to like replace RT. It's just that it has this history. It goes back a decade. So when you go to like look up a ticket, you can look up tickets that are like it. And so when you move over, you're going to lose that. And so no one in the group wants to lose that. So it's, and RT also has a command line interface, and such is the nature of my group, we all actually read our tickets from the command line. So none of us actually log into that thing. It's all in the terminal. So. All right, great. Well, thanks for coming. It's great to kick off the research computing version of the ABRF session. I don't know that they usually do these, so. It's cool to see structural biology cores and research computing making some inroads amongst the omics.